In this video, we'll learn about the structure of DNA. All right, so let's take a look at the components of DNA. And it's important to remember at this point that DNA is one of the group of molecules called nucleic acids. And nucleic acids are built from subunits called nucleotides. So each nucleotide is made up of three components. They have a base, and they have a 5-carbon sugar, and they have a phosphate group. So in this illustration here, you have the base, and then you have the 5-carbon sugar, and then you have the phosphate group. Now, there are 4 different nitrogenous bases. So when I say nitrogenous base, I'm referring to this portion of the illustration that we called the base just a moment ago. It is, in fact, a nitrogenous base. So you'll hear me say that a lot, so I just want to make sure that you were clear that the nitrogenous base wasn't something different. It is the base. So again, there are 4 of these different nitrogenous bases that we find in DNA. Now, the reason that I only called it a base on the previous image is because all 4 of these versions of the nucleotides contain a different type of nitrogenous base. So we just say base in general. But now we're going to be specific as to which nitrogenous base is contained in each of these nucleotides. So the different nitrogenous bases are adenine, guanine, cytosine, and thymine. And they are typically represented by a singular letter, basically the letter that word starts with. So adenine is known as the letter A, guanine is known as the letter G, so forth and so on. So how does the structure of adenine and guanine differ from the structure of cytosine and thymine? Well, the answer to that question is that adenine and thymine have 2 rings, and cytosine and thymine only have 1. And that's going to be significant in a moment as we continue to explore this molecule. Okay, so as we look at a molecule of DNA, it has a lot of these nucleotides. Now, one single nucleotide is this portion right here. So again, the phosphate group, the 5-carbon sugar, and the nitrogenous base, whatever that nitrogenous base happens to be. Now, in this case, it consists of adenine. Okay. Now, there would be another nucleotide here that has the guanine nitrogenous base, and then a cytosine, and then a thymine. So we have in total 4 nucleotides here. Now, of course, that's not an entire molecule of DNA. A molecule of DNA, it literally has millions of these nitrogenous bases. Now, something else that we need to be aware of is the way that these nucleotides come together and kind of bond with each other in order to create the full DNA molecule. Now, the way that they create and the way that they kind of stay together is through these covalent bonds that occur between the various nucleotides. So you see the arrow pointing here, and you'll find if you look at each of the nucleotides, you've got one right here, you've got another covalent bond here, another covalent bond here. Now, there's not another nitrogenous base below this one, below the thymine, so you don't see another bond down here. Those bonds only occur when those nucleotides are close enough to each other to have that bond. So it's important to note, too, where that bond is happening. And where it's happening is at the sugar of one of the nucleotides and the phosphate group of the next one. In this case, if we start at the bottom, we've got the phosphate connected to the sugar, this phosphate connected to this sugar, and this phosphate connected to this sugar. Okay. Now, there is this rule that we have found to be very accurate, and it was discovered that the nucleotides behave in this manner by a gentleman named Erwin Chargaff. Okay, Erwin Chargaff. Now, he discovered that the percentages of adenine, the A, and the percentages of thymine bases are almost equal in any sample of DNA that you look at. And he also found that to be true of the guanine and cytosine. Basically, there was the same number of adenine nucleotides as there were thymine nucleotides. And with guanine and cytosine, there are about the same number of nucleotides of each of those. Now, the number of guanine and cytosine was not necessarily equal to the amount of adenine and thymine that were found in a sample of DNA. That could be different. But the adenine and thymine tended to be pretty equal, and the guanine and cytosine tended to be pretty equal. So we get this rule that is A equals T and C equals G. Okay, and we're going to find that these nucleotides, the A and the T, will pair up with each other, and the G and C will pair up with each other. That'll make sense in a moment as we kind of put this molecule together like a puzzle. So speaking of putting this molecule together as a puzzle, the key to really understanding the DNA molecule was to try to figure out the shape of this thing. And that was very difficult for scientists in the early 19 to mid 1900s. But Rosalind Franklin, this fantastic scientist, she used a technique called X-ray diffraction in order to study the structure of the DNA molecule. So what she did is she purified a large amount of DNA, and then she stretched the fibers to make them all parallel. And then she aimed an X-ray beam at the sample and recorded the scattering pattern that was made on the X-ray film. Well, the results of her work revealed some very important clues to the structure of DNA. First, the X-shaped pattern that she saw in the DNA X-ray showed that the strands of DNA are twisted around each other like coils of a spring. This is a shape that we call a helix. And then the angle of the X also suggested something else, that there were two strands to the molecule. So other clues suggested that the nitrogenous bases of the nucleotides were actually on the inside of the molecule. So again, what she was able to determine was that DNA is a helix, that there were two strands to the molecule, and that the nitrogenous bases found in this molecule were always near the center of the molecule. Now, about the same time that Rosalind Franklin was doing her work with the X-ray diffraction images, Watson and Crick were also trying to figure out what the shape of this DNA double helix was, so that they could learn more about the molecule. Now, they approached the problem of DNA structure by trying to build three-dimensional models, and they made them out of cardboard and wire. What they were trying to do was make it possible from a biochemical point of view to kind of see this molecule, basing it on the known properties of DNA. So they twisted and they stretched the models in various ways, but everything they did, even their best efforts, really did not explain DNA's properties. But then, when they found Franklin's work in 1953, they realized how they needed to fix their model to make it work. Now, The clues in Franklin's X-ray pattern are really what enabled Watson and Crick to build a model that explained the specific structure and the properties of DNA. The twisted double helix model explains Chargaff's rule of base pairing, the A with the T and the C with the G that we talked about a little bit ago. And it also explained how the two strands of DNA were being held together. Now, let me ask you this. If a twisted ladder is used as a model of DNA, which parts of the DNA molecule correspond to the sides of the ladder? So imagine a ladder and then you twist it. What would it look like? And what parts of the ladder would be the parts that correspond to the sides of the ladder? Well, if you're thinking the phosphate group and the 5-carbon sugar make sense for being the outside of the ladder, you would be correct, and they form what's called the backbone of the molecule. So then, which parts are left of the DNA molecule to correspond to the rungs of the ladder, if we're going to carry that analogy through to its completion? Well, the rungs of the ladder would be representative of the nitrogenous base pairs that meet in the middle and connect A to T and C to G. If you need a little additional help in grasping the structure of DNA, there are lots and lots of videos, and we'll probably watch some in live class that you can look at, and they will help you understand this a bit better. But we'll probably take a look at some of those in live class as well. But just keep in mind that if you were to take the double helix and untwist it, it would look like a ladder where the rungs of the ladder would be the nitrogenous bases and the outside of the ladder, which we call the backbone of the DNA molecule, would be the phosphates and sugar molecules. Now, one of the surprising aspects of the double helix model is that the two strands of DNA run in opposite directions. And this arrangement of strands is described as anti-parallel. And this arrangement enables the nitrogenous bases on both strands to come into contact at the center of the molecule. Okay, now you see the arrows here pointing in opposite directions, one pointing up and one pointing down. Now, again, as I mentioned, the two strands of DNA are held together by hydrogen bonds between the nitrogenous bases, adenine and thymine, or between the guanine and cytosine. So, which part of the nucleotide do the two strands of the DNA molecule join? Well, they join at these bases. Okay, so one strand is on one side, the other strand is on the other side, and in the center is where the nitrogenous bases form a hydrogen bond. Now, keep in mind also that the strand itself is being held together by those covalent bonds between the sugar and the phosphate of each nucleotide. Okay, but the two strands are being held together. So the nucleotides are held together by the covalent bonds. The two strands are held together by the nitrogenous bases with hydrogen bonds. Okay, very important. Watson and Crick had realized that hydrogen bonds between the two strands would explain the tight fit, but that hydrogen bonds would form only between A and T and between C and G. This fit between bases is called base pairings. So the two strands of DNA are held together by hydrogen bonds between the nitrogenous bases adenine, or A, and thymine, and they're held together by the hydrogen bonds between guanine and cytosine. So, what is the relationship between the base pairing rules and the evidence for Chargaff's rule? Remember, Chargaff's rule stated that there were the same basic amounts of adenine and thymine, and there were the same amounts of guanine and cytosine. So, how does this relationship between the base pairing rules, the A to T and the G to C, how does that relate to Chargaff's rule? Well, if A only pairs with T and C only pairs with G, then for each base of a particular kind, there would have to be one of its partner bases. Therefore, they would be in about the same percentage of volume.